Good morning. I'm Gretchen Sassenrath. I'd like to talk to you today about measuring soil biology, and more importantly, how we can use that to improve production and profitability. Soil health has been defined as the continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. Soil health we want soil health to relate to productive capacity. The functions of healthy soil include sustaining life, most typically of crop plants and animals, but also the soil microorganisms. It provides stability and support, nutrient cycling, filtering and buffering, and water cycling. The productive capacity is the bushels per acre, or more importantly, the dollars per acre, the net return of our production. Nutrient cycling and water cycling are two of the most critical things for crop production. In the soil, if we look at the soil as a storage system, we then want the nutrients and water to become available to the plant when the plant needs it. And when it's finished, to go back and put the excess back into the storage for later use. Soil is composed of three major factors. The physical components we're all familiar with are the sand, silt, and clay components of the soil. The chemical components make up the pH, the nutrients, and the water. The biological components are quite diverse and includes plants, bacteria, fungi, nematodes, arthropods, insects, earthworms, and animals. These three factors interact in unique ways to support plant production and improve soil health. The soil biological components include the plants, including the roots, the bacteria and fungi, the protozoa, nematodes, arthropods, earthworms, and animals. All of these function within the soil and add to the soil function. From the plants, we see the plant roots, and in particular, these very fine root hairs really increase the absorption of water and nutrients in the soil, transferring that to the plant. They're very important. The root hairs are very important because they can get into the soil matrix and extract nutrients and water for plant growth. Overall, in an estimate of 1 teaspoon of soil, we have 100 million to 1 billion bacterial cells, several yards of fungal hyphae in an agricultural soils. It will be much more than that in the less disturbed prairie and forest soils. Hundreds of thousands of protozoa. We've got nematodes as well. Some of them are positive and some of them are negative. And then per square foot of soil, we'll have many arthropods and also earthworms, depending on the particular conditions, whether it's arid, semi-arid, or deciduous. The soil bacteria are an important component, and we're very familiar with the nitrogen-fixing bacteria. These are gram-negative. They're symbiotic. They associate very tightly with the plants, called mutualists. In exchange for sugars from the plant, these bacteria convert atmospheric nitrogen to a form that's usable by the plant. Then we have the nitrifying bacteria. They convert ammonia to nitrite and nitrate, which are more usable by the plants, but also are more easily leached. Then we have the denitrifying bacteria. They convert nitrate to nitrogen of nitrous oxide, and these tend to be anaerobic, so they're working under conditions of no oxygen. The actinomycetes are gram-positive bacteria. They are decomposers, and they decompose especially hard to decompose substances, such as chitin, which are found in insect skeletons, and cellulose. Fungi include the saprophytic fungi, which again are decomposers that convert dead organic material into useful sugars and organic acids. The mycorrhizal fungi, which are mutualists, similar to the bacteria. They colonize plant roots, and again, in exchange for sugars from the plants, they provide phosphorus, nitrogen, micronutrients, and sometimes water to the plant. Pathogenic fungi cause major production losses. These include verticillium, pythium, rhizoctonium, phytophthora, fusarium, and so on. There are also parasitic fungi, such as the nematode trapping fungi. They parasitize nematodes. Others can act as biocontrol of insects. Why is all of this important? Well, we cannot change the physical makeup of our soil. God gifted us with sand, silt, and clay in a particular ratio, and that's pretty much what we end up farming. We can influence the chemistry of the soil, most notably by the fertilizers that we apply and sometimes water through irrigation. But we can greatly influence the biology of the soil with different management practices. And managing the biology can improve the overall interaction between the physical and chemical components to improve the productive capacity of the soil. So how do we measure soil health? Well, first of all, as I mentioned, we have microbes. That's the microbial community. These microbes make enzymes. Those enzymes take a particular substrate, organic compound or whatever, insect skeleton, whatever it is, and use that enzyme to transform it into a particular product. That product is then used by plants for growth and production. We don't harvest the product of the microbial activity. We harvest the product of the plants that rely on the products from the soil microbiome. We can measure the amount of microbes or the type, the amount of substrate, the amount of enzymes or their activity, and the amount of product. In all of these things then, let's start with talking about how we measure the amount of substrate that microbes use. I'm going to talk a lot about our soils in Parsons, and we have this is a typical profile for a Parsons silt loam. You see, we've got about 10 inches of pretty good soil, this nice black stuff that we like that. Then we get a very high clay content. And clay content can get up to 50% in this layer. And then that clay pan or that clay layer persists deep into the soil profile. And you can see the change in color as we go deeper in the soil. So this is a fairly typical soil profile in southeast Kansas. If we look at the change in clay content again, we'll have a very high clay lens. And in these slides, I'm going to show you, these are from 3 different production types. Conventional till, a hay meadow, which had been in long-term grass production, and then no-till. But you can see for all 3 of these, we've got a fairly typical clay content. It increases very quickly to a very high clay content within the soil profile. We also have a change in the soil pH. It's fairly good at the top, and it decreases as you go lower in the soil profile, and it's fairly consistent for the 3 production types. The carbon content in the soil is an important substrate for microbes. You can see in the hay meadow, we have quite a lot more soil, especially in the upper 4 inches of soil. We've got a little bit more carbon in the no-till than we do in the conventional till. The carbon content decreases very rapidly as you go into the soil profile. Similarly, the soil nitrogen content is much higher on the hay meadow than it is on the no-till or on the conventional till. Again, nitrogen is a substrate for certain soil microbes. The phosphorus content shows a similar decrease, being very high at the top on the surface of the soil and decreases very quickly as you go deeper into the soil profile. By 12 inches, there's no difference between any of the production practices. Potassium shows a little bit of a change here. We've got high content of potassium in the upper surface that decreases, and then it increases as we get into the clay pan. So things change with potassium when it's in the clay layer. This is a similar profile as well with sulfur. We see sulfur builds up in the clay layer. By measuring this substrate within the soil, we can get this kind of information. So this is one field that we measured, and we took measurements at 0 to 3 inches, 3 to 6, 6 to 12, or 12 to 18. You can see we start out in the silt loam at the very surface of the soil that very quickly, for this particular field, very quickly goes to the heavier clay, the much heavy clay, silty clay. Even in some instances of 3 to 6 inches, we're in the heavy clay pan. And by 6 to 12 inches, a good bit of the soil is the heavy clay pan. The results from a typical soil nutrient analysis give us this kind of breakdown. You can see the black dots are the 0 to 3 inch level. And most of the soil, most of the fields, we did 8 different locations in the field. Most of the locations in the field have good pH levels, but there are some areas of the field that could use a little bit of liming. Much lower at 3 to 6 inches, we have a much lower pH in this field. Similarly, for the phosphorus recommendation, we see most of the field, especially at the 0 to 3 inch range, has enough phosphorus. It's plenty good fertilizer with phosphorus. The average from 0 to 6, though, shows that some of the locations in the field could use additional phosphorus added. And we can do this for all the other nutrients as well, the nitrogen, the potassium, and so on, and get these kind of results. If we look at a different field, this one has a lot more consistency throughout the field. We see none of the layers are in the high clay. That means it has not eroded as much. So this is a much better field, if you will. And we see in this case, again, we've got a distribution of the pH and the phosphorus requirements. But in this case, almost the entire field is fine with the phosphorus level until we get to the very lower levels, the 6 to 12 inches. And again, like I say, we could do this kind of nutrient analysis for all the different parts of the field and look at the different things. These kind of soil test interpretations with the fertilizer recommendations for the different soil types, Dorovar does this, has done it quite well. And we know quite a bit of how nutrient results for the field and nutrient applications are going to relate to productivity in bushels per acre or dollars per acre. That's fairly straightforward. Like I say, that's measuring the stocks or the nutrients that these different microbes would use. Getting back to how do we measure soil health, let's look at how we can measure microbes. And again, we can measure the amount and the type. One of the most ubiquitous measurements for soil microbial activity is called the PLFA, which stands for phospholipid fatty acid. The microbes are made up of a cell wall that has, or cell membrane that has a particular phospholipid bilayer in it. This type is particular for each type of microbe present. The different bacteria and the different fungi have different PLFA compositions. What we measure in the PLFA is actually the microbial membranes. This is a kind of report you get out when you measure soil microbial community analysis. It returns the total biomass, the diversity of your particular sample, and then it breaks it out into functional groups. How much gram-positive actinomycetes you have, gram-negative, the rhizobia or nitrogen fixers, the total fungi, the arbuscular fungi, the saprophytes, all these different groups, protozoa undifferentiated. Quite a detailed analysis. These samples are around $80 per sample, fairly hefty price. We can see again, looking back at our soil profile, we have a huge microbial biomass. Note this break in the axis here. We're up at 120 grams per gram, or nanomoles PLFA per gram of soil. For the hay meadow, it drops off quickly, but it always stays well above the microbial biomass in the hay meadow than it does in the no-till or in the conventional till. And in this case, we can see a difference between the no-till and the conventional till, but they do drop off fairly quickly as you go deeper into the soil. We've also measured some differences in biomass for the different types between no-till and tilled. And again, total bacteria, undifferentiated, gram-positive, gram-negative, actinomycetes, total fungi, saprophytes, and arbuscular mycorrhizae all change with tillage practice. So that gives us an estimate of, or how we can measure the microbes and amount and type. We can also look at the enzymes. And in this case, we can look at two factors, the amount of enzymes or the activity. One of the frustrations we have is the activity is very difficult to measure in situ. In other words, the enzyme might be present in the soil, but it might not be active in the soil. When we take out the enzyme and measure it in the assay it in the lab, we put substrate in and put perfect conditions in, so we get kind of an artificial or optimal activity. It's not the actual activity that we see in the soil or that may be occurring in the soil. But again, we can look at beta-D-glucosidase. This degrades cellulose, so it's a carbon forming enzyme. Very highly active in the hay meadow. Decreases as we decrease lower. There's a little bit of a bump in the clay pan, but really not much. Slight increase for the no-till over the conventional till. But again, they all drop off very quickly after we get past the surface. Another enzyme that's present in the soil is an acid phosphatase, which is used to access phosphorus or to make phosphorus. Again, we see a similar, very rapid decrease in activity, which much greater activity in the hay meadow than in the no-till or the conventional till. In this case, we do see an increase in the acid phosphatase activity within the clay layer, at least for the hay meadow and the no-till. Again, the N-acetylglucosamine shows a much bigger decrease in activity in the clay layer, which is a different profile than we saw in some of the others. This enzyme is used to access nitrogen. Again, much more activity in the hay meadow than in the no-till. It is an interesting difference in the profile is that it's more active within the clay layer. And we've looked at this enzyme activity and also active carbon going through the whole crop cycle. So we start with corn. We see the enzyme activity increase quite a bit for wheat production, and it kind of mellows out a little bit, comes up evenly with soybean production, and then stays constant with fallow. So there's a shift or a change in enzyme activity depending on the crop, depending on the time of year, depending on the crop stage, and all these things change how the soil biology is functioning. This is another important point, though, to make too, is that depending on when you measure, you're going to see a very different change in enzyme activities and nutrient status. So how do we measure soil health? We've looked at the microbes, the substrates. Let me show you another method of measuring activity. And these are things that are a lot easier to measure than the lab measurements that I have presented so far. One thing that you could do is the Solvita labile aminohumic nitrogen test called the SLAN test. It takes a set amount of soil and puts a little paddle in it, and the paddle changes color depending on how much nitrogen is being released in the soil. The refills, each one of these paddles are about $10 a piece. There's also the Solvita CO2 burst test. That one relies on a set amount of soil. You add a known amount of water to it and measure the CO2 burst after the test. It starts out as a blue paddle and changes color to a yellow paddle if there's a lot of activity. These give you an instantaneous measure of how much activity, enzyme activity there is in the soil. One of the frustrations of these, even though they're much more reasonable as far as price goes, they have to be fairly consistent as far as how much soil is put in and how much water is put in. Another test that can be done is the Haney soil health analysis. This test relies on three major factors. One is the soil respiration, which is the CO2 burst test. Also the organic carbon and nitrogen. And then they have a special H3A extract that measures different components. This is the kind of output you get from the Haney test. These tests are about $45 a piece for each sample. Again, you're getting a lot of the substrate measurements. You've got total nitrogen, nitrate, phosphorus, all of those things are being measured. And then you also get the CO2 burst, gives you a soil health calculation and some suggestions for it. So those are a lot of how we measure these different things. We can measure the microbes. We can measure the enzymes. We can measure the substrate. How do these measurements relate to productivity? And again, this product that we're developing here is the product of the microbial activity. It's not corn or beans. It's not the product from plants that we're harvesting. One of the biggest frustrations in all of these measurements, it's that research has shown that all these soil health measurements do not relate to yield. So it's very difficult to translate microbial activity to dollars per acre. The thing that this group of researchers found is that the health measure that most closely correlates or indicates yield was the CO2 burst test. So how do we improve soil health? Where's the magic genie? These are some things that we cannot do. I hope you understand enough now of how the soil microbiomes work that you'll understand why we can't do these things. We cannot add enzymes alone. If you add an enzyme, it will translate or change the substrate into the product, and the plant may be able to use that. The problem with pouring in a bottle of enzymes is the enzymes are not living. They are finite. Once they run out, they cannot make more of themselves. So once the enzymes go through the reaction, they're pretty much dead. Another thing we cannot do, we can't add microbes without substrate. The microbes will make the enzyme, but most of the time the microbes make the enzyme in response to the presence of the substrate. Without any substrate, the microbes are not going to make the enzyme. So you won't get any of the product you need to improve or boost plant productivity. Another thing we cannot do is add substrate without microbes. Again, the microbe makes the enzyme, but in this case, without the microbe, you're not going to have the enzyme. So again, if you boost hugely the organic matter, that's a substrate for the microbes, but you don't have microbes a lot around, the substrate is going to sit there. An interesting thing, when you first convert to no-till, you'll end up with a lot of residue on the surface of the field. That will change over a couple of years very quickly, because the microbes will come when the substrate is there. So how do we use all this to improve soil health? And really focusing on bushels per acre or dollars per acre. How do we improve nutrient cycling and water cycling? Well, gonna be boring. 5 principles of soil health. Keep it covered. Soil armor. Reduces erosion, moderates temperature, reduces compaction, suppresses weed growth, and provides habitat. By having the soil covered, it really improves the overall soil structure and function. Minimize soil disturbance. Go to a no-till or at least a reduced till situation. Increase the diversity within your production. Rotations are good. Cover crops are better because it puts even more variability into your system than corn, beans, and wheat. And provide a continual living plant or root, which is cover crops will allow that. Also integrate livestock. Some of the things that I've looked at before, simply the cost of tillage. You have equipment and fuel cost. This is $14 per tillage pass. You have a time cost of about 8 acres per hour. Soil, you lose in the best circumstances 2.9 tons per acre per year, which results in a decrease in your productive capacity of the field. You have nutrient losses with tillage of about $15 an acre. You lose the water holding capacity of the soil. You lose productivity of about 23 cents per acre. The yearly cost of tillage is then about $29 per acre per tillage pass. If you make more than one tillage pass, you increase each time you go over the field. Tillage costs directly. Another big factor is the soil chemistry. Every 1% increase in organic matter increases soil available water more than 20,000 gallons per acre. You need about 4,000 gallons of water to grow 1 bushel of corn. So if you're aiming for 100 bushels of corn per acre, you need about 400,000 gallons of water per acre per year, or about 1.2 acre feet of water each year. Again, remembering 1% increase in organic matter increases soil available water more than 20,000 gallons. This is the water use curve for corn in a Silt loam in Labette, Kansas. You can see at about the highest growing point, we need about 3/10 of an inch of water per day. So we need about an inch of water every 3 or 4 days. Improving the organic matter directly improves the soil water holding capacity. Tilling also directly improves the water holding capacity of the soil. These are some measurements I did in Labette County. This is the water that's in the soil for a conventional till, a soil that has been 5 years in no-till, and a soil that's been in long-term no-till. It was actually longer than 10 years. Big increase in water available in the soil just by going to no-till. And that a lot of it is the tillage improves the soil structure, improves the organic matter, and you have more water available in the soil for the plant growth. Important consideration for soil moisture. This is a well-watered soil, and this is a temperature. This is the air temperature in the crop. And you see we've got a nice balmy 87.5 degree day. This is full sun. This is sunlight here. We've got a full, bright, sunny day. This is the canopy temperature. That's the canopy, that's the temperature of the leaves in the crop. You can see they've got a good 5-degree cooling because this is a well-watered crop. Compare that with a water-stressed crop. Same day, same temperature. In this case, these plants are 10 degrees warmer than the air temperature. They are not able to cool. So there's about a 15-degree temperature difference between a well-watered crop and a water-stressed crop. And that temperature difference is going to directly impact the yield and the productivity of this crop. So water is very important. The more water you hold in the soil, the more water is available for the plant. Cover crops are another excellent way of improving the overall soil health. One of the things that I've done, supported by Kansas Soybean Commission, was look at cover crops for disease control. This is a high glucosinolate mustard cover crop. A lot of folks claim all mustards are high glucosinolate. Some of them are, some of them are not. In Southeast Kansas, we have a lot of charcoal rot, and you can see that it's extreme in this case. This guy has so much infection from charcoal rot. There's almost no root structure there. But this gray charcoal stuff is the charcoal rot that has gotten into the vasculature and is basically strangling the plant, which is why the roots have died. You can also see it a little bit easier, actually, even this one here. You see these gray areas in the stem that are the charcoal rot fungus. What we did is planted this mustard seed in the biological control and chemical control, and we see a decrease in the colony forming units, which are the bacteria that cause or the fungus, excuse me, that causes the charcoal rot. The control had quite a lot of charcoal rot. The biological with the mustard seed had a greatly reduced activity of the colony forming units of the macrophamina. And again, looking at different ways of managing that mustard. This is the control. They had no mustard. If we disked the mustard, we actually lost a lot of the control. This is the difference between post and pre. You can see the best way of managing the cover crop is to roll it. So minimal disturbance of the cover crop, rolling it down, flattening it, and then planting the soybeans into that had the best control of the charcoal rot. Again, that showed how the cover crops can be used to control soil-borne diseases. These high glucosinolate mustards have also been suggested to control nematodes. So there's a lot of things that we can do with cover crops to change the pathology of soil-borne diseases. Another thing we could do is use cover crops for weed control. It's a lot of research has shown, and this is some demonstration that we have in Parsons for different cover crop mixes. And the weed density is this line here. The cover crop treatment was different amounts of biomass of cover crop. The fallow, even with a herbicide, had very high, the highest weed density. Grays of radish did not grow well. The forage collards also did not grow well, had a much lower biomass than the other cover crops. Wheat was a very good cover crop, covered well, had a much lower weed density. And we've shown this for 2 years now. The more cover crop biomass you have, the fewer weeds you have. Another big factor in how to improve soil health is feed them and they will come. If you increase the organic matter in the soil, it increases the food sources needed by the microbial communities, which then mine more nutrients, soil nutrients for the plants. If you don't have a food source for the microbes, they will not come and they will not make the enzymes that create the products that the plants then need. So you have to be sure and remember to feed the microbes. Again, 5 principles of soil health: keep it covered, minimize soil disturbance. You'll save money by stopping tilling and have a really huge improvement in the soil tilth and the health in the field. Increase the diversity, provide a continual living root, both of which could be done with cover crops, and integrate livestock. And there is an opportunity for livestock to graze the cover crops. Some of the factors, the important factors to consider: it's easier to preserve topsoil than to make it. It takes a very long time to make topsoil. Poor soil health compromises the productive capacity of the soil, and building soil health is a long-term commitment. You're going to see changes in the first year or two. You'll really start to notice it after 3 to 5 years. Cover crops can improve soil health and profitability. Cover crops can increase soil organic matter, increasing microbial activity for better water and nutrient cycling. And there's a selection tool, Midwest Cover Crop Selection, to select cover crops for the results desired. With that, I'd like to thank everybody who was involved in this research. Jerry Shao did a lot of the enzyme assays and studies. Vladimir Alikhan has looked at erosion. Stacy Kaleza and Mark Mathis were involved in the erosion studies. And Lonnie Mengarelli has done the lion's share of the actual field work. I'd also like to thank the sponsors: Kansas Soybean Commission, Kansas Crop Improvement, NRCS, and National Science Foundation. With that, I'll take any questions. Thank you.